Ladies and gentlemen, you're all very welcome to the 2021 Darwin Day lecture. Thank you all for so much for joining us, albeit in different circumstances to the norm, of course. We're celebrating the birthday of Charles Darwin, which is actually tomorrow on February 12th. And for the last few years, this event has filled the Robert Emmet Theatre on Trinity's campus, about 160 people each time. And judging by where the numbers are going, I'd imagine by the time we actually get started, we could be looking at something similar as well in 2021, which is great and wonderful to see. So thanks all for giving up part of your evening to be here. My name's Eamon Murphy. I'm events coordinator with the Humanist Association of Ireland. I'm going to be as brief as possible with some little bits of housekeeping before passing over to David McConnell, who will give the opening address. First thing is to say that this event is being recorded. I believe it's customary to give due warning in that regard. Secondly, I'd ask that you please keep your microphones muted for the duration of the evening, unless you're called on to speak or ask a question towards the end, in order to kind of somewhat mitigate the impersonal nature of having to hold these events remotely. We much preferred it when it comes to Q&As in these sessions, as will be the case after that more people should be able to ask their own questions rather than typing them into a box. So the main thing about that, though, is that we need to trust you then in the I beg your pardon, in the meeting function, not the webinar function, to keep people's microphones on mute. Please don't take offense if I do mute you on my control panel here. You can leave your cameras on if you wish, but I recommend selecting speaker view rather than gallery view so that you can get a better glimpse of David and then Aoife when they're speaking. And you can control that with the little functionality in the top right-hand corner of your screen. As I've alluded to, once Aoife is finished speaking, we're going to have a Q&A. To draw your attention to your desire to speak during the Q&A by virtually raising your hand, what you do is you click on the participants icon at the bottom of your Zoom screen, and a tab will appear on the right showing all the attendees. At the bottom of that, there's a button that says raise hand. So I'll remind you how to do that later, but yeah, again, you do it by clicking on participants and then the raise hand button at the bottom of the right-hand screen. Now, it's tradition to preface every Humanist Association meeting with a short explanation of what humanism actually is. So very quickly, humanism is an ethical life stance. It takes a rational outlook towards life, attaching primary importance to human matters rather than those of a divine or supernatural nature. And humanists aim to live meaningful and ethical lives without being guided by religion. Essentially, we aim to be good without God. And our key values are reason, equality, and compassion. Now, any questions that you have on the HAI or about humanism, either you can Google the Amsterdam Humanist Declaration, or you can give me an email at events@humanism.ie. And with all that out of the way, without any further ado, it has been tradition for the last number of years that our honorary president has given the opening address of this event. So it's always something that I look forward to almost as much as the lecture itself. And I'm pleased that he's agreed to maintain the tradition even in lockdown, especially as he's a man who himself knows a thing or two about genetics. So I'll pass you over now to the honorary president of the HAI, Professor David McConnell.
Thank you very much, Eamon, and good evening to everybody. There seems to be a little bit of over talk. I don't know whether I'm the only one hearing it. In any case, off we go. So it's an enormous pleasure always to take part in the Darwin Day ceremony lecture. And it's a very special pleasure for me today because, or this evening, that's because Aoife McLysaght is going to give the talk. Not the first time that she's given it, but this happens to be special for two reasons. Many, but I'll mention two. One, it is the 150th anniversary of the publication of The Descent of Man by Darwin, 1871. It's also the 20th anniversary of the publication of the draft sequence of the Human Genome sequence. And Aoife, at the time, a graduate student, was a co-author on that paper, one of the milestones in the history of genetics. So we're celebrating two anniversaries, Aoife. Now, if I'm not going to give an address really, because I'll just say a few words. I really want to listen to Aoife, and I think the rest of you should too. Just reminding you that in 1859, Darwin published The Origin of Species. Certainly one of the 2 or 3 most influential books, I would say, in the history of science. And he introduced his theory of natural selection. And he was deeply aware of the impact the book was likely to have, because he knew, of course, that his theory of evolution by natural selection applied to people as much as it did to any other organisms. But in the entire book, he only referred to that question once. A famous sentence, he said, "Light will be thrown on the origin of man and his history." By the way, he was a broad-minded person. He was using the conventions of his and man, and typical of the time, which I suppose many of us did until maybe 30 years ago. Now, he followed this in 1871, and if you think about it, 1859 to 1871, it's only 12 years later, he published one of his other most influential books, The Descent of Man. And it had a subtitle, not so much a subtitle because it came in 2 parts, and Selection in Relation to Sex. So it was an extraordinary book, but he noted around about the time, he said, everyone is talking about it without being shocked. And on to say, proof of the increasing liberality of England. So whereas The Origin of Species had created a furore, The Descent of Man was greeted with, I won't say equanimity, but by that time, the thinking, reading public in Britain, well, he says England, he wasn't a little Englander, by the way, but he meant Britain and Ireland, indeed. The thinking and reading public at the time were no longer shocked by the idea of evolution. A very large percentage of thoughtful people had already accepted the idea, A, that evolution had occurred, and a large number of them had reckoned that he probably had the right answer, that it occurred by natural selection. And by the way, sexual selection is a branch, if you like, of natural selection. So there was no shock. And as time has gone by, of course, his great ideas, and there were so many of them, in The Origin of Species and otherwise expanded in his many other books, including in The Descent of Man, so many of them have turned out to be correct. And he had enormous insight. There was, let's be said, there were huge problems. If you read The Origin of Species, you'll find that he's really rather careful to mention the large number of difficulties with the theory of evolution by natural selection. One of them, which bothered him all, is, well, for a long time, but in the end, he got over it. He tried to imagine how an eye could evolve over great periods of time during the evolution of animals. How could such an incredible organ as the eye appear? By the time he gets to 1871, he's thereabouts, he's writing to say that he's got over that problem. He just thinks that eyes started off as something very, 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 very simple and got more and more and more complicated as the millions of years rolled by. But the peacock's tail bothered him intensely because why would evolution lead to something so outrageously, apparently useless, even damaging and difficult as a peacock's tail? That all gets sorted out, by the way, in his theory of sexual selection. Now, it goes on. I mean, Darwin is such a rich, rich, rich thinker. I'll give you one other thing that occurred to me in thinking about The Descent of Man, which I have to confess I have not read, but in it, he makes, and maybe Aoife will talk about this, but he is quite clear that all people on the planet are members of the same species. And remember, we're talking about the middle to late 19th century. Slavery was not very far in the past. Indeed, it was to some extent still in the present. And of course, the echoes are so strong even today. But he was in absolutely no doubt that all people on the planet were members of the same species. And further, it's lovely to read a little bit about his life. He started off life as a medical student in Edinburgh. He spent 2 years there. And there he met a man called John Edmonston, who was a Negro brought from Guyana by a Scottish business person, I don't know, adventurer, given his freedom in Scotland, and he became a teacher of taxidermy in the medical school at Edinburgh. And Darwin learned from him and came to know him and delight in him and wrote about him, about what a splendid person he was and how intelligent he was. So quite early on, he was only 17 or 18 years of age at this time, by the way, he had met a wonderful Black person and was in no doubt at the time, of course, about that person's enormous value and dignity as a human being. So when he traveled around the world on the voyage of the Beagle, and he met people of many different races and so on, he was already well primed to think of people of all human being of the same species. Now, he also, in The Descent of Man, talks about social evolution. He realized that as human civilization had developed, that social evolution had begun to have a very big effect on the evolution of people. And that, of course, was something that his cousin, John Galton, thought and wrote and talked about, and is much maligned in some ways. But Darwin nails his colors to the mast in no uncertain terms, that even if social evolution has given us the power to, as it were, thwart natural selection, a person like myself who is blind as a bat would not have lasted very long. He nevertheless was quite clear that we had a great duty and indeed fundamental belief in the value of everybody to look after everybody in society. So one thing which comes through whenever I read Darwin, is the great humanity of the man. Now, that said, my main duty this evening is to say something about Aoife. And I'm lucky enough to have known her longer than almost any of you. And although I don't know the entire audience tonight, but I was lucky enough to meet her when she was a second year junior officer student in Trinity. She took a problem course, which I had set. She got 100%. And that summer I asked her, would she help me to set the exam for the next year? And this is one of the great pleasures of being a teacher, when you find students who are much smarter than you are. She went on to a splendid career in genetics, a wonderful degree, a wonderful PhD under Ken Wolfe. She is the academic granddaughter of Paul Sharp, who invented the theory, not the theory, but the field of molecular evolution in Ireland. And in her work, she has shown just wonderful, I would say, imagination and genius, just terrific qualities. And we were very lucky. She went off to the University of California at Irvine, spent a year there, but then she came home. And she has been with us since, I think, 2003, and has contributed absolutely enormously to not only genetics worldwide, but I would say thrown herself heart and soul into the teaching and fostering of genetics in every possible way in Trinity. If I were to characterize her work or focus of her work in one or two words, she's been very interested in showing how genes have been added to genomes. So Ken Wolfe had shown that yeast at a particular time, that is the ordinary Saccharomyces cerevisiae, the ordinary brewer's yeast, had suddenly doubled its number of chromosomes. Aoife went on to show that vertebrates had not just doubled, but quadrupled their number of chromosomes. So we are, in a funny kind of way, a tetraploid species, but that's not quite the right way of saying it. And then I remember the time when she actually discovered that since humans had separated from the great apes, she discovered new genes had been added, and she showed how they had been added. Absolutely wonderful, wonderful work. So much fun to see it being discovered and seeing the cleverness with which she identified these new genes. So she's been in Trinity for the last 18 years as a member of staff, highly valued. She's written a very large number of scientific papers, many of them really characterized by, I think I used the word genius again, and cited a very, very large number of times. Absolutely terrific. She's a brilliant teacher. And that leads me just to mention the joy of being a teacher. The beauty of being a teacher is that every year you find new students. And they sort of keep you up to the mark. But I want to mention that Aoife had a teacher. I hope he might be watching tonight. I asked him to join in, Dr. Nick Fruin at St. Andrew's College, a brilliant secondary school biology teacher who turned out 3, 4, or 5 wonderful students for the genetics department, Aoife being one of them. And she has shown an enormous commitment to teaching, not just inside college, but outside. I mean, I don't know of any other academic colleague in the country who has appeared at Electric Picnic. She's given TEDx talks. She's talked at the Royal Institution Christmas lectures. These are enormous indications, measures of her brilliance as a teacher. Known, I want to say, from, and you can see it from those comments, known worldwide for her capacity to inspire people and to educate people. Professionally, she was awarded a President of Ireland Young Researchers Award in 2005. She has won research grants from the European Research Council. That is an extraordinary achievement in any scientific field. A starter grant in 2013. And then not many starters go on to consolidate and get a consolidator grant, which she has at the moment, starting in 2018. And then to have had the honor of being asked to give the JBS Haldane lecture to the Genetical Society. And then the accolade, perhaps, of which, well, we are very, very proud. She became the president of the Society for Molecular Biology and Evolution. That's the international society, essentially based in the United States. But in 2017, she became the president of that society. Now, many of you will be aware, I hope you have been, that she has contributed enormously in founding the Irish Society for giving independent advice to the government on the COVID crisis. She's been a great pillar of that organization. I should also say that she is now my successor as the head of the genetics department. So it is with enormous pleasure and indeed anticipation, Aoife, that I look forward to your lecture tonight. Thank you very much for being willing to do it. Thank you.
Thanks, David. I think I'm going to be a letdown after such a wonderful introduction. I can't possibly live up to that. But thank you so much. And if Dr. Fruin is in the audience, that'd be fabulous. I definitely credit him with starting everything in terms of inspiring me to think about science and to think like a scientist. I think it was always an extraordinary example of how teaching isn't just delivering information, but making you think. And I really think that's what it is to be a scientist. And it started in a classroom setting. I have a copy of Descent of Man here, which is pilfered from the genetics department library. It has like the Department of Genetics and a little code, but I didn't nick it. You did, David, because this was in one of the boxes you gave me when you were moving house and you needed to downsize your bookshelves. So I'm innocent. I'm only a beneficiary of the theft. Anyway, so yeah, I was looking through this and taking some bits out of it. So I'm going to talk today about this book because it is the anniversary, and I am going to just give me a second. I will share my screen. Okay, so Eamon, I'm not permitted to share my screen right now. Could I do that, please? Okay, yeah, it's letting me do it now. Okay, so everybody should be able to see that. So, and I'm just going to, there should be a way for me to make a little laser pointer. Yes, okay, so if I want to point at things. So as David mentioned, there's an anniversary this week, not this month, 150 years since the publication of the Descent of Man. So this is not a first edition, so I don't think it's a valuable one that you pilfered, David, but it's still lovely to have and to hold. And so what I thought I would do today is I was going to try and talk about this book in a modern context. And it's really quite a challenge because the book includes so much. So I'm going to talk through some of the ideas. There's a few places where it's really interesting to see how right Darwin was, you know, given the information he had at the time. But there are other things where you can clearly see the information just wasn't there. Most notably, I think they just, at that time, didn't have the idea of the gene or DNA or whatever was the heritable material. And there's bits in the book where he sounds a bit like Lamarck, which is funny because we usually set up Darwin's ideas and Lamarck's ideas as the opposite. But he does mention how he has a section where he's talking about, you know, certain people, you know, using their muscles more, and maybe their children will also develop to have more muscles and things like that, and bigger muscles, or particular physical features that are useful in their daily life. So it does sound a bit like Lamarckism, but he possibly meant it a bit slightly differently than that, or at least it could work slightly differently than that. So as David mentioned, the book has two titles, The Descent of Man and Selection in Relation to Sex. So it's actually, I think, more than two titles. It's basically two books, because the selection in relation to sex is a whole topic unto itself. And in that section, he's not really talking about humans anymore. He's talking about all kinds of other animals. So I dispense of this one quickly because I decided that I couldn't do everything. It would be very, very interesting and exciting to have a talk about sexual selection all by itself. But instead, I'll just really briefly say it is discussed, and it is basically a special case of natural selection. So natural selection, as I think everybody here probably attends these lectures every year. So maybe you're all far too knowledgeable for the kind of things I'm talking about today. But natural selection, of course, being just that idea of there are variations which are heritable variations, and those variations that help you do better in your life. So survive better, get better, you know, get your food better, and all of those things will be the ones that get passed on, because you're going to be a reproductive success story. And then sexual selection is a special case of this, because mostly in natural selection, those things that help us survive better and have a better chance of passing on our genes, it's because they make us healthier, or because they make us better at something, you know, better at avoiding a predator, or better at finding your food, or all of these kind of activities or these features we can think of. And then when you get to something like the peacock's tail, it doesn't seem to fit those criteria, because it's this enormous showy target sign that the animal has displayed. So, and not only a big target sign that makes it so ostentatiously visible, but also probably makes it easier to catch, because it's a big thing you can catch onto and take hold of. And so it was this puzzle, in a sense, in terms of evolution, because how could something that's so obviously an impediment have evolved? And that's where Darwin came up with the idea, well, there's a special case of natural selection which is all, it's in the eye of the beholder. So this beautiful tail evolved because the peahen likes it and for no other reason. So the only advantage, the unique advantage that the peacock's tail gives it is that the peahen likes it. And so he has a better chance of finding a mate. And of course, this is a very elaborate, extraordinary structure, and it didn't happen in one mutation to go from like a more typical bird's tail to a peacock's tail. But it's a case of what would be described as runaway natural selection. So the female has a slight preference for a bigger tail, and then you've got slightly bigger tailed peacocks, and then one of them has a mutation and his tail is a bit bigger, and that one does even better at attracting a mate, and so on until you've got the most extraordinary, showy, ostentatious, catch me if you can sign on the peacock. These things you tend to see in males in nature. So when you're talking about animals, you tend to see the showy animal is the male. And that's because typically in nature, it's the females who are doing all the choosing. And so the females, you'll know this if you look at any birds in your garden, the females tend to be more plain and they have more plain plumage, and the males are the showy ones. This also happens in fish, for example, in guppy fish. The males are quite colored and showy looking, but the females are much more plain. And this is another example where it's an advantage for the male to be colorful because it helps him attract a mate, but for the female, there's no advantage. So it's only a disadvantage if she was colorful, because then she's more visible to a predator. So she gets all the pain, but none of the gain. So this is a thing we see throughout, it's something we see in general throughout nature. But I'm just going to talk about the first part of the book, which is all to do with human evolution. And it has a few different sections on this. So I have here just the chapter headings, and you can see the general topic. So the first two chapters, I'm putting them together, and essentially what he's starting out here. So as David mentioned, the origin of species just had that very brief mention of human evolution. Light will be shed. So he waited a few years and he's shedding that light now. And you can see where he's starting his arguments from, because he is arguing in many ways from first principles, you know, that he's trying to say, first of all, you know, that humans aren't separate from all of nature. So humans are animals, just like any other animal, really. And he also then talks about, so he's talking about here various structures that are shared. And in the second chapter, he's taking on this kind of following on the same idea. He's talking about inheritance, and especially the laws of variation are the same in man as in the lower animals. So basically, you know, genetics is the language of life. He didn't know the words genetics and he didn't know how genetics works, but this is why evolution is such a powerful concept and such an important concept, because it unites all of the life on this planet, and it does it through the fact that that genetics and genes are the same everywhere. So his basic arguments, he was looking for similarities in form across different animals. He also talked about rudiments, so of ancestral forms, so certain forms that were just still hanging around in a rudimentary form, which is evidence of some of the shared ancestry. And then of course that the laws of variation are the same. So this idea of similarities in form is probably well known to everybody. And it was his contemporary Owen who had a drawing a bit like this one, looking at the forelimbs of various vertebrates. So even though these vertebrates are very different, like so humans have a hand at the end of the forelimb, cats have this paw, whale has a fin, and a bat has a wing. So these are, all of these different vertebrates are extraordinarily different. But when you look at them, you can see that the same bone structure is there essentially, even though it's been stretched and distorted in the different animals. So in this picture, the colors are showing you the equivalent bones. So you've got one big bone, one big bone, one big bone, one big bone, followed by two smaller bones. So you got 1, 2, 1, 2, 1, 2, 1, 2, followed by a whole lot. And so these are the yellow, green, and blue. So you see the same structure in all of them. So even though these have been changed, you can see that similarity. And so this was the kind of argument that was being used at the time. So we can see this in our bones, but then we can see the same pattern essentially in our genes. So the same logic applies. So here you see a slightly different version of the long, big blue bone, slightly different versions of this orange bone that you can recognize they're the same even though they're changed. And we can do the same with genes as well. So here I have some gene sequences, some DNA sequences. I must have cut off the label. So this was human, bat, armadillo, and horse, if my memory serves correctly. And so you see here the letters. So this is just the way we write DNA. I think most people are familiar with this by now, GCs, A's, and T's. And so you've got across the top line is human, bat, armadillo, horse, and then it's human, bat, armadillo, horse. It's just the sequence is looping over the pages, around the page. And what you can see here is the dark coloring shows you when it's the same across all 4 of them. The slightly paler is when it's the same in 3, and then the pale blue is if it's just in 2 out of the 4. So you don't have to be very clever with maths to see that these are really very much the same across these 4 different animals. So even though I've taken DNA sequence from these 4 different animals, you can see the huge similarity between them. So this is like seeing that the bones are the same across the different limbs. And in fact, I picked this gene because this particular gene is HOXA13, I think, or 14, and it is responsible for forelimb development. So I picked the gene that is actually driving the development of the front limb, the forelimb. So we see it's the same gene. I don't think anybody would dispute that this looks like the same thing in these genomes from very different vertebrates, but with a few little changes. So we can see bits where there's differences. So we can see, just like we see in the bones, we see variations of the same form, and it's showing the ancestry. You see this very, very clearly in the genes. So the common ancestry is really, really clear. So these DNA sequences, like every DNA sequence, was inherited from the parents and the parents and the parents. So there's an unbroken chain going back of inheritance right back to the common ancestors, and the common ancestors of all of these different vertebrates, even though they've got very different forelimb structures. And so this DNA sequence has remained almost undisturbed, some very small changes over most of vertebrate evolution. And part of the reason this works is what Darwin was referring to when he was talking about the laws of heredity being the same. And so, of course, we all know by now that we all have DNA, every living thing except for viruses. We all know about viruses now, especially RNA viruses. So some viruses have DNA, but some viruses only have an RNA genome. But this is a common thread all through all of life. And the way a gene works is the same everywhere as well. So what I'm showing you here is basically, it's a eukaryotic cell. So it's not a bacteria, it's not a virus, but the principles are the same in bacteria. So you've got the DNA, which is really, really long, contains all the genes. This can be transcribed and processed into an mRNA, which everybody now knows about as well, because we've been talking about mRNA vaccines over the last year. And the mRNA is a messenger. So the M stands for messenger. So the RNA is just a version, slightly different chemical version of the DNA. This gets sent out into the main body of the cell where it gets converted into a protein. So we know about this now because this is the basis for some of the vaccines that have been produced against the SARS-CoV-2 coronavirus. So the Moderna and the Pfizer vaccines are both using mRNA technology, which is taking advantage of the fact that this is the common language of biology. So you can take a sequence from a virus, which is what they did. You can then just read it in your computer. You can synthesize it, and you can then insert, and what the vaccine does is it delivers an mRNA, which codes for, instead of any human protein, codes for the spike protein on the outside of the coronavirus. So when this gets taken up in the cell, this bit of cellular machinery, the ribosome, comes along, it reads it, it follows the instructions, because those instructions are universal. It doesn't matter that it came from, that it's supposed to be a virus sequence. It's a universal language of biology. So it builds the correct protein, building one amino acid at a time. And the protein it builds is a spike protein of the coronavirus. So that gets presented to the immune system, and it learns to recognize it as foreign, and that's how that vaccine works. So this works because of this common language of biology. So it doesn't. So in this book, Darwin is arguing that the laws of biology and the laws of variation apply also to humans. They didn't know about viruses yet, but it applies. The same laws apply there. And we're also learning about virus evolution, aren't we, lately, seeing how the virus is changing when it's given the opportunity, when there are selection pressures, that it gets a competitive advantage. And the competitive advantage, of course, one of them is making more copies of itself by being more transmissible. So we're seeing that there's lessons in molecular biology and genetics and evolution happening worldwide in the last year or so. So in the book then, Darwin then comes to the conclusion that humans are animals just like any other animals. So man is liable to numerous, slight and diversified variations, which are induced by the same general causes, are governed and transmitted in accordance with the same general laws as in the lower animals. So it seems that when he first wrote The Origin of Species, he was, the idea appeared controversial. He was perhaps hesitant to apply it to humans, but here he makes this very clear statement. And as David said, it wasn't even too controversial by then. So that is already good news. So when we think about those, so the next thing that Darwin started to think about was development and the laws of development. So here I'm showing you these pictures of the head of a fly, a Drosophila melanogaster fruit fly. So this is a normal head, as it normally should look. And you see there are these little antennae as well. So these are the eyes, the big fly eyes. And these are the mouth parts and the antennae coming out. And then we have this monstrous mutant. It isn't a small mutant, it's an extraordinarily enormous mutant, because instead of having a small change, it actually has legs growing where the antennae should be. So it's a monster. And the weird thing here is that this monster came about by a very small genetic change. So in this experiment they were inducing small genetic changes, and the small genetic change created a monster. So it was a huge, big step in terms of what could, like the difference that this mutation caused. But this was really powerful, because it was the first indication that there were universal master genes in terms of controlling body development, that there is a gene that a small change in one gene can make a whole structure grow in the wrong place. And these were labeled Hox genes, so you might have heard about them before, but this was one of the first discoveries. So this particular gene was called antennapedia, because it was making the leg grow where the antennae should be. And so when these discoveries, so in the same idea as Darwin thinking about, how he can apply, how these laws apply, do they apply to humans and do they apply to other animals equally? There was this challenge in thinking about the whole process of development and how the genes can encode the instructions for a complex structure to grow. So this is from 1984. And I'm including this statement just because it's in hindsight embarrassingly wrong. I wouldn't be, maybe I'm picking on this person, but here we have, he says that embryologists are confronted with an ensemble of unique phenomena. We cannot expect to discover a general theory of development. Rather, we're faced with a near infinitude of particulars which have to be sorted out case by case. So this particular person was very pessimistic that there could be any commonality in development between different animals. And if you think about something like the fly, so not only does this fly look so radically different from us, for example, or fish, or pick any other animal you like, it also goes through, like, its development is very weird as well, because it's first born as a larva, and it spends some of its time in a larval state, and it goes through a metamorphosis before it becomes the adult form, a bit like a caterpillar becomes a butterfly. The idea that there might be anything in common between us and something so peculiar was quite strange, but the ink wasn't quite dry on this statement when it was discovered that the similarities in development exist really very, very deeply. So this is the way Darwin was looking at it. So this picture is from, there's not very many pictures in The Descent of Man, but this is from The Descent of Man, and it's showing different embryos. So on the top, we have a human embryo, and on the bottom, we have a dog embryo. And what he's pointing out here is the similarities in form at these various stages of development. And this is a well-known phenomenon and well recognized, that as you look through development, you see different stages which are very similar to each other, and sometimes almost difficult to tell which is going to be which. So we have these similarities that you can observe during embryology. And then when we look at what's happening with the genes that lay out the body plan, what you can see is at the top here, we have an embryo or a larval form of, I think it's probably the embryo here, of fly. And at the bottom, I think we can recognize the mouse. And we have these genes, which are these Hox genes that I mentioned already, like the one that makes, when it's mutated, makes the antenna grow out of the wrong place. So that's that one there. ANTP is antennapedia. So that's the one that got mutated to get the antenna, the legs growing where the antenna should be. And if you look, this is the length of the growing embryo. And here we have in a row, the genes are represented. So the line is just the DNA strand, and the boxes are just representing the genes. And these have all been given colors. And actually the color of the gene is represented up here as well, which is the region where that gene operates. So in laying out the body plan in the fly, you can see these genes are expressed in particular domains, which makes the body plan come out the way it comes out. So a leg grows here because the correct, so the antennapedia, the red, is growing here where the legs are supposed to grow. So when you get antennapedia expressed, you get the legs. If you express antennapedia in the wrong place, a leg is going to grow out of the wrong place. So we see this similarity in the embryos, and we also see the similarity in the genes. So here we have what's happening in the fly, but then you have the same genes that are also in a mouse and in us and in all vertebrates. We have actually, instead of just one string, one line with the colored boxes on them, we have 4. And that's because of one of the things that David mentioned in his introduction, that there was a doubling and a doubling again in the origins of vertebrates. So 1 became 2 and 2 became 4. And that's how we end up with 4 copies. But we have the same genes, although in more copies, and we see the same phenomenon, that the genes at one end get expressed in one end of the body, and the genes at the other end get expressed in the other end of the body. So this statement that there'd be no general laws of development turned out to be extremely wrong, because not only are there general laws, but the genes are the same. So you see that the genes that make a fly body develop correctly are the same as the genes that make a mouse body, and indeed a human body, given how dramatically different they are. And it even goes further than that, because in all kinds of different animals with all kinds of strange structures, we see the same thing happening. So again, we see the genes are in a row, and the colors are showing you both the correspondences, well, the lining up shows you the correspondences between the different animals, and the colors show you where the genes are turned on in the different growing animals. And you see here, okay, we're in with the tetrapods, so we look like something like that at an embryonic stage. We can see in fish, we have the same. We see the color, the rainbow colors, all in order. We talked about the fly already, and then these polychaetes, these funny insects down here, these ones that aren't, they're just outside the vertebrates and are quite different. And then you get these things like urchins, which also have these genes. And so these genes are really very, very common throughout life of different animals. And we see that there's this commonality. So even these extraordinarily different body plans, very, very different, like this one has this radial symmetry, and these extraordinarily different body plans all have the same fundamental genetics under them. So these laws certainly apply and are certainly universal. So another thing that Darwin was looking at then was what he was calling rudiments. So he had, this is a picture he has, and he's obsessed with this little bit of cartilage or little detail that some people have in their ear. And if you look at it in, so he has it here in The Descent of Man, but if you look at it in later textbooks, they call it Darwin's something. So this little structure is named after Darwin now. And what he believed this to be was, if you imagine a pointed ear of a different primate, that this was what's the rudiment of the point having kind of slightly folded in. So not everybody has this little structure in their ear, but some people do. And so he considered this to be a rudiment of the ancestral pointed ear that's just become folded in. He wasn't totally, he wasn't like going to bet his house on this. He said, you know, there's a chance this could be just an anomaly of cartilage, but I think he was pretty confident on this. And then, of course, if we look again at the developing human embryo, you can see a very obvious tail as well, which at this stage is even more prominent than the limbs. And so that's another example of some rudiment we have, you know, something that is not there in the final structure and is not needed, not used by us anymore, but it's a legacy of the shared ancestry. And of course, this is our coccyx in an adult form. So we can see these things. So Darwin was looking at these structures because this is what was available to him. But we also see this in the genes in terms of genetics. So this picture here is a representation of the first ever clinical trial. So where there were a case control experiment on a medical intervention, which was done for scurvy. So these are sailors on a ship, and sailors on a ship could frequently suffer from scurvy, and it wasn't known what was causing the scurvy. And Lind was I'm actually not sure that he must have been a doctor, but he decided to try different treatments for the scurvy. And he'd actually, one of the things he tried was lemon juice. And the account that I read of it was actually, he didn't really think that lemon juice was a credible treatment. He just put it in anyway, kind of like as a, it wasn't his favored hypothesis when he started. But of course, the individuals who received lemon juice were the ones who did well, because we know, they didn't know then, but we know that the lemon juice contained vitamin C, and scurvy is caused by a vitamin C deficiency. So we need vitamin C. A vitamin, by definition, is something that we must obtain in our diet, but we only need in small quantities. So we need vitamin C, we only need a little bit of it, but we need it because we can't make it inside our own bodies. So vitamin D is not a true vitamin by that definition, because we can make it in our own bodies in sunlight. But we used to be able to make, ancestrally, we could have made we could have made vitamin C, which is to say that not our human ancestors, but like vertebrate ancestors. So there's this gene, which has this catchy name, L-gulano-gamma-lactone oxidase, which is the last step. This gene carries out the very final step in the vitamin C biosynthetic pathway. So when you have this gene, you can make vitamin C from other ingredients, and you can do it in your body because it's a biochemical process to make the vitamin C inside your body. And this is this gene. So it's broken up here into 12 parts. That's just because the gene is broken up in the genome. And this green coloring is just showing you that this is kind of how it's human. What you find is you don't find the gene, you essentially find a rudiment of the gene. You find where you find that kind of broken structure in the same way that we have a hint of a tail, but not a real tail. We have a hint of this gene, but we don't really have it. So these yellowy shaded boxes are actually deleted. They're missing in the human genome. So this gene is only partial, and the partial gene doesn't work. So it is not a complete gene. So that is because we are missing this, we can no longer make vitamin C inside our own bodies. And independently, but similarly, the guinea pig cannot make vitamin C. So if anybody has a pet guinea pig, I guess you need to give it some vitamin C in its diet. But so what happened? Why did we lose this gene, especially considering without vitamin C, we suffer quite badly? Well, there is no, we don't know 100% for sure why we lost this gene, but the very plausible explanation is that actually we had at some point our ancestors, so this is lost in other great apes as well. So it wasn't just in humans. So in the primate ancestry, we were possibly living with a rich dietary source of vitamin C. Whatever we were eating habitually had a lot of vitamin C. So it doesn't, your survival doesn't depend on how you get your vitamin C. So if you get your vitamin C because you're eating it, or you get your vitamin C because you're making it inside your own body, there's no difference. All that matters is that you got vitamin C and you don't get sick. So if you then are in a member of a population or a species that's eating these fruits, for example, commonly, and you get a mutation, one individual gets a mutation that deletes the first chunk of this gene so that it no longer works anymore, that individual isn't going to suffer in a natural selection sense. They're going to pass on their genes as well as anybody else because they're not missing vitamin C. They don't make it in their own body, but they're not missing it. So there's no disadvantage to losing the gene in that context. And because there was no disadvantage to losing it, there was nothing that was preserving it. So it wasn't being kept. So it's this kind of idea, use it or lose it. So there's no preservation force. So because natural selection, a lot of what natural selection does is it preserves things, keeps things as they are. And that's why when I showed you that gene sequence of the Hox gene that's determining the forelimb, we saw that really strong similarity across the different animals. That's because natural selection is preserving it. When a mutation occurs, those individuals aren't surviving as well as the others. So that mutation gets rejected by natural selection. Here, the mutation didn't get rejected because the individuals did just fine. So we see this, so it's the same principle as Darwin was talking about, but we can see it now in a genetic context. We see these leftovers. So he pretty convincingly showed that there's this really strong connection between humans and all of the other animals. So he says, we have given man a pedigree of prodigious length, but not, it may be said, of noble quality. So anybody who wanted to claim they were better than the animals or superior and all of that doesn't really have a leg to stand on. So I think it's a lovely quote. So then in the next section of the book, I'm not going to talk for too long about it because it's harder, but he's talking about the comparison of the mental powers. So we do know though that when we consider ourselves superior, as we are wont to do, and we think it's all to do with our big brains. And so evolution of the human brain is a very interesting phenomenon, and it's a very interesting thing to understand. And so when we think about it, when we look at it, just to briefly show you here in this paper, what they've done is they're looking at these 4 different primates. So we've got bonobo and chimpanzee, which are quite closely related to each other. And we've got human and we've got macaque. And so we carry those colors over here. So the macaque monkey is green, the human is red, and then we got the bonobo is the purple, and the chimpanzee is the blue. And so this is just an analysis of the genetic variation and the variation of genes expressed in the brain. And one thing you can see is that you can see this difference. You can see that the bonobo and the chimpanzee are close to each other, and so they have less difference, not much difference. But then the macaque is clearly different and the human is clearly different. And you can think about these in terms of where they're expressed in the brain. But the important thing is that we find the same genes. Again, we find the same genes are present in the brains of these different primates. So it's not, we don't find there's no magic key, there's no single thing that we can find that says this is what makes the human brain different from other brains. This is what makes the human brain bigger, what makes it capable of the more abstract reasoning, or all of these things that we might think are important. And so we don't find a single genetic cause. And so what we find is small differences, but we find the differences, but they are individually small, but maybe with a large cumulative effect. And Darwin talked about this because he was talking about it then. He said, you know, it's not that we're separate. We're not a totally different kind of thing. We're just a bit more in one direction. So he's talking about then the difference in mind between man and the higher animals, great as it is, certainly is one of degree and not of kind. And he talks about this idea a fair bit in the book, where he talks about, is this a difference of degree or is it a difference of kind? Is it that you're just, you know, is this something that's totally, entirely novel, or is it just edging one way or another? He also goes on to, I think one of the interesting things he talks about, again, I think he was probably very much interested in getting the readers to know their place and not feel too superior, because he then talks about how ants have such an incredible intelligence. So ants are capable of complex communication. They carry out these tasks and all of this thing, and they are tiny, and therefore their brain is tiny. And he says something along the lines of gram for gram or ounce for ounce, whatever he would have used, that the ant brain has probably the most spectacular brain on the planet. Okay, so then the last sections of the descent of man, as opposed to the sexual selection, is where Darwin then talks about, you know, the origins of humans. So he spends the first bit talking about, you know, putting that, you know, we are part of nature and the same laws apply to us. But then he starts thinking about, you know, the origins of humans. So he thinks about, where did we come from? You know, how did we arise? How are different humans related to each other? And what is the significance or the value of our differences? Are these differences important? So this is where, as David mentioned in his introduction, he has this whole chapter which he describes as on the races of man. And it, I think it's quite, you know, when you're used to thinking about things in modern language, because he is talking about whether he's asking the question, can we be considered all one species, or are we different species? So I think it's not, you know, it's uncomfortable to read that, but the way he approaches it is quite interesting, because he says, if you were a naturalist and you came along and you were looking at these creatures for the first time, with some of those creatures being, you know, one of those creatures being himself, how would you analyze them, and what are the tools you would use to analyze them, and how would you decide how they're related or how they're different? And so he talks about how if you wanted to categorize, you know, there are some features that you can see are different if you wanted to, but then there are so many things that are constant across all of them. But then I think importantly, even on the features that you might think are different, and of course, skin color being the most immediately obvious one, you cannot put them, and this is, I think, the most convincing point, which shows where, like, as David said, like where his conclusions lay, is you can't put them into categories, because it's more like a continuum. So if you think of skin color as an example, you can go from very, very dark to very, very pale skin, but you don't have categories inside that. You'd basically got all the shades in between as well. And so he kind of says, well, you can talk about 3 races or 5 races, or maybe you talk about 20 or 300, and that tells you that it's not sensible at all if you can't even describe what difference you're trying to look at. But in terms of where we come from and we think about it, we can, of course, these days look at this from a genetic point of view, and there's been extraordinary work done on this in recent years, because the availability of DNA sequencing and how cheap DNA sequencing is these days has made a huge difference. So this is one way of looking at it, which is down a microscope. And these are, so it's chromosome 1, they're a little bit bent and twisted, chromosome 2, 3, 4. And the human chromosomes are numbered in order of size, just except for the X and the Y, which are, of course, special because they're to do with sex determination. But the other ones are numbered in terms of size. And so you can see them down the chromosome here. And we've got two chromosome 1s, 2 chromosome 2s, etc. But if you just count one of each, we've got 3.2 billion letters of DNA or so. And of course, we represent the letters as A, C, T, G. And if you pick any 2 individuals at random in the world, unrelated individuals, so not ones with a close relationship, but they're going to be different on average in 1 in 1,000 letters. So that's the same no matter which people you pick. So if you pick somebody from Africa and somebody from South America, or if you pick somebody from East Asia and somebody from Europe, you're going to find this difference is around about the same. So that's our first interesting observation, that we are a very young species, and the diversity we have and the relationships are all pretty equal. You know, we find this 1 in 1,000 difference more or less for anybody. And so we can think, though, we can still think about it. So most of the differences like that are, you know, quite constant or consistent across the world, but we still can, there is a residual small amount of difference that we can do, which can be used to trace geography. So we can use it to trace geography as well. So this picture here is the Laetoli Footprints in Africa, which are fossilized footprints showing upright or bipedal, walking. So these footprints have been studied then, and they are the first known example of bipedal walking. So they're considered a very important discovery in terms of understanding human evolution. And we can think about genomic footprints and not only physical footprints, you know, in terms of tracing how humans have moved around the world. And so we can do this with a kind of very simple principle, which I'll explain here. So if we imagine we have this starting population here, so, and here each circle is an individual, and the slightly different colors are just showing you the genetic variation. So this is a population of different individuals, and there is genetic variation. And this is a reasonably big population in this pretend example. So it can carry a fair amount of diversity. So if you have just 2 people in your population that you're studying, that's obviously got a limited carrying capacity for genetic variation. You've got 2 copies of each chromosome in each person. So maximum is that you got a version on your 1 copy and a different version on your 2nd copy, and the same in the other individual. So you got an absolute cap on the amount of genetic diversity that can be carried at any one position in the genome. So you've got more people, it's just got more capacity to carry more variation. So this big-ish population has been around for a while, and this diversity has accumulated over time in this population. So this is our starting population. And let's just imagine a scenario where some individuals from this population migrate and found a new population somewhere else. But it's only a small number of individuals, so 3 in this pretend example. So by definition, they can't carry all of the genetic diversity that existed in this big population. So they're just carrying a subset of the variation. So in this case, a green, orange, and blue variant. And then that can grow up over time. And we see this new population has got bigger, but so far it hasn't gained any new diversity. So we see these 2 populations, and this one, it can be recognized as a subset of the diversity of this one. And then the same process can happen again. Some individuals from here found a new population, and so on. So we can see this effect. So we can see what's these founders, the ones who found a new population in a new place. Each time you're sampling the diversity. So we get a lot of diversity here. You take a sample that founds this new population and a sample that founds that one. And we basically do this analysis globally on humans, and you can see this type of pattern. And so what you find is that the area of the world with the greatest amount of diversity is Africa. So in Africa, we find the largest amount of genetic diversity. And then as you step out from Africa, you can see this sampling effect. So as you step out from Africa, you see a sample of this diversity is here, and then go left, go right. There's a sample here, and then sample, and you see this sampling effect. So this is really useful because it allows us then retrace the genomic footprints, which are actually tracing literal footprints, because people, of course, would have walked as they migrated around the world. So genetically, it's absolutely unambiguous that our species originated in Africa and that we migrated from there gradually over time. And then we can look then in a more modern context at, looking at the diversity, the genetic diversity globally. And this is a really famous image in science circles, which is probably a little niche, but there you go. But this paper is it's from 2008, so it is a good few years old now, but it is very much a classic paper, and this type of analysis has been repeated over and over on different human populations. So what they did here was they got DNA samples from individuals around Europe, and they did a statistical analysis on these DNA sequences that they had taken. So as I said before, most of the genome is really, so most of the interesting parts of the genome is really conserved. We see a lot of mostly similarity across all of the globe, and this average 1 in 1,000 still applies. And then there's a small amount then of the other variation that we can see this other kind of residual signal in. And so what they did was they did this genetic analysis, which essentially is a statistical analysis that just sorts out the differences. So this is PC1 and PC2. It's a principal component analysis in case anybody's really into their statistics. But a principal component is just a statistical measure, which is kind of saying what is the axis, if you like, that finds the most variation, and then the second most and the third most and the fourth most, and you can go on to lots and lots of principal components. But here we're just spreading it out in 2D space, statistically decided. So the information that went into this analysis was just the genetic information, but then they colored their data points in this graph afterwards, according to the country where they had taken the DNA sample. So if you look at this, you can see here's Ireland with Great Britain beside it, even Scotland in the north of Great Britain, just like in the map. Then we've got France here with Spain and Portugal down below it. So here we've got the Atlantic coast of Europe, Italy projecting out into the Mediterranean. And then you can see Germany and Denmark, Sweden and Norway, Finland, Poland, Czechoslovakia. So the genetic variation is mirroring the map of Europe extraordinarily well. And when you think about that, this analysis was done entirely blind to the origins of the samples. The samples were all treated the same, and this coloring in only happened at the end to illustrate the point. So the genetic analysis showing is really powerful in showing these little these differences and these geographic origins or geographic relationships, actually. So this is not comparing to the past, it's only comparing to the present. So it's saying that the Italians, are like each other, basically, and the Irish are like each other. It's not saying where things came from. And this I will emphasize mainly because sometimes this type of thing gets misused, that this is a really tiny amount of the variation that gives us this kind of signal. It's a small bit of the genetic diversity that is not, like most of the genetic diversity is global and you find it all over the place. But this shows, I think, some of what's really interesting and powerful that you can do in terms of understanding human genetic diversity and origins. But when we look at the general, I would say the kind of diversity we see in the genome is mostly inconsequential. And I mean totally inconsequential, as in nothing happens. So you have that 3.2 billion letters of your DNA, only about 2% of that is really genes, and the rest of it is just stuff. And most of the time when that stuff changes, nothing happens. So we can use it when we're doing these genetic analyses, and that's probably the kind of thing we're seeing here. There are variations in the stuff, just the packing, and not the actual genes, but they're useful, they're informative if you're interested in history. Sometimes they have an effect, but they're not that important. An example of a variation that has an effect, but we don't think is terribly important would be, for example, your blood group alleles. So this picture is showing you the frequency of the A allele. So you have the A, B, O blood groups. So you can, and somebody will be, so there's the A, B, and O alleles that give you the A blood group, the AB blood group, the B blood group, and the O blood group. So this is just showing you A. And the darker the color, the dark blue is the highest frequency, white is the lowest, and then we've got this yellowy red. And so you can see there's quite dramatic variation in the frequency of this A blood group allele across the world. Like it's really, really low in South America, for example. It's really high in this bit here of North America, really high here in Finland. And then you see intermediate in other places. So this is a genetic variation. It's got a global distribution that looks interesting, but it doesn't change your health status. It doesn't, not that we know of, give you any advantage or disadvantage. So it has an effect, but it's not important. But sometimes we have variations that are really interesting. So the classic one of these that you've probably heard about before is evolution of resistance to malaria. And so there's a very interesting pattern if you look at the map of Africa, and this dark green is showing you where there is malaria infection, and this orange is showing you where you find a particular variation of one of your hemoglobin genes. So one of your alpha and beta globin together make up your hemoglobin, and there's a variation, there's a mutation, different version of the beta globin that is common in parts of the world where you find malaria and utterly rare everywhere else. And this mutation causes your red blood cells to form this weird shape, which looks a bit like a sickle. So it's called sickle cell. And So you can see here, that's the normal shape of a red blood cell, and this is the sickle. So it turns out, if you have sickle cell, if you carry one normal copy of the beta globin and one sickle cell copy, you're going to have some of your red blood cells are going to be malformed like this into a sickle. And if you have two of the sickle forms, you're going to have all of your blood malformed like that, and I think you'd be very, very ill. But if you have one malformed like that, you have some of your cells are one of your copies of the gene is the sickle, then only some of your red blood cells will be shaped like this. And so it turns out, the reason you get sick with sickle cell is those red blood cells, when they're in that shape, don't work so well. You can get a lot of pain, and you get so a lot of other effects, and then quite anemic as well. But when you carry only one copy of that gene, you're anemic, you have some consequences. And if you're living in Europe, you just have anemia, and it's no advantage at all. But if you're living in a malarial area, it turns out having these sickle cells helps you clear the malarial parasite out of your body better, and so you don't get malaria. You're resistant, at least resistant to malaria. And so it's partly, it seems to be because these sickle cells are targeted for recycling more efficiently. And so the malaria parasite gets picked up and recycled out. And so this is a case where this is an advantage. This is a mutation that is regional, geographically regional. This allele is common in this part of the world and rare everywhere, because it gives an advantage in that part of the world and not in other places. But there's another interesting localized genetic adaptation, which appears to be an adaptation to diving. So in Southeast Asia, there's this group of people who are known as the Bajau, I'm possibly mispronouncing that, who dive essentially for their food collections. They go fishing by diving. And this is a long established tradition. And so there has been over many generations selection in this population, apparently, for being better divers and being able to hold their breath for longer. And what we see is in this population, there's a mutation in this gene, and that mutation results in a larger spleen, and a larger spleen helps these individuals retain more oxygen from in their body for longer. So that's a really interesting example of where we have some of these interesting and useful differences globally. And then this picture summarizes a whole pile of these, because there are loads of them. So this gene EDAR1 is associated with thick hair. You can see there's a little key here at the bottom. So people of East Asian ancestry tend to have beautiful, thick, strong hair, and it's due to a variation in this particular gene. We see again, these are variations in genes, adaptations which give resistance to malaria. So that's a mosquito there, which carries the malarial parasite. So HBB is hemoglobin beta, the beta globin that we mentioned already, and there's some other known there. There are examples that are to do with dietary adaptations. So there's adaptation to dairy diet, which is basically lactase persistence. So milk is baby food. And in most animals, after weaning, the animals can't digest milk anymore because they don't normally eat it. But in humans of various groups of humans, so some humans of European descent, so we have it up here in Europe as well, but also in the Middle East and Africa, it's occurred independently, that there has been this adaptation to eating milk in adulthood. So basically that enzyme lactase keeps being produced even into adulthood, which means you can digest the lactose sugars in milk. If you don't have lactase, you're going to be lactose intolerant. And when you drink a glass of milk, you're going to get a very upset tummy. But then we see other examples. So adaptation to cold, adaptation to altitude. This EPAS1 is very interesting because this particular adaptation was possibly acquired from Denisovans. So this adaptation exists in a region of the genome which has actually been in Tibetans, is found in Tibetans and in the Denisovan, which is an archaic human, a bit like Neanderthal. And so we see these different parts of the world, we've got these different adaptations to diet, to disease resistance, so and adaptations. The hair in this particular case, the most plausible explanation for that is that it is an example of perhaps sexual selection, that it was just a preference, the beautiful glossy hair. So that's it. That's my summary, my big kind of whistle-stop tour through Darwin's first part of the Descent of Man, because I could do a whole lecture on each chapter, I think, because there's so much that we now know. But I hope that was interesting. I hope it wasn't boring for those of you who've seen many Darwin Day lectures before. But it's a tough crowd in this place. You're all far too well informed, and I'm really happy to hang about and chat and take any questions or whatever you like.
Aoife, thanks so much. That was fantastic. I can assure you that there was nobody bored, and the 140 people that were here till the end is a good sign of. Well, this way, I think that David's compliments on your skills as a teacher are clearly well founded. So I think the traditional way of expressing appreciation in these circumstances when you're muted is to.
Okay. Well, thanks very much,
everybody doing that. You were speaking there for an hour. Do you want to?
Was I? Sorry.
Take a break.
I'm okay. I mean, I thought I was going to be half an hour there. I talk for, and there you go. Sorry about that. Is anybody still?
There's certainly no need to apologize. Don't worry about that. I guess just to remind people, I can't actually see this in my screen because I'm the meeting organizer or whatever, but I believe, and I tested this at the weekend, if you click on participants, a tab should appear on the right-hand side of your screen, and there'll be a raise hand button at the bottom of that.
Yeah. There's another way of doing that as well, Eamon. Across the bottom, you have reactions. And oh no, actually, sorry, it's not. It's only in the webinar version. So you're right. Go ahead. Okay. Yeah, it's there. I see it though. I see it in the raise hand. But I just see other questions. Oh, somebody's asking me if Pfizer or Moderna are better than Oxford, Russian or Chinese. They're all different. So, I mean, this is my own fault for mentioning the vaccines. But so the Pfizer and the Moderna are very good. But like one of the big advantages of the Oxford is the ease of distribution. So it's, one with a slightly lower efficacy, but that is easier to get to more of the population, might overall be the better vaccine in terms of vaccination, the protection is, there's an individual protection potentially, but the better protection is the population protection. So the better thing is that it gets to as many people as possible. Somebody else is asking about the vitamin C. Sorry, am I moderating? Am I taking your job, Eamon?
What I was going to do was just unmute the people, just to kind of begin some kind of more conversational tone.
So people asking questions in the chat.
Lee Hogan Kerrigan, you can unmute yourself there if you want to ask your question on vitamin C, so people understand the context if they're not looking in the chat box.
Hi, Aoife.
Hiya.
I was asking about, you were talking about vitamin C and the fact that because humans didn't need it because there was enough source around them, it kind of got dropped somewhere along the way. But what about the fact that, we actually do need it and most of us are not getting enough for various reasons? Would it be a possibility to re-evolve? That's all. Just what, how evolution's still going on, I'm assuming.
Yeah. So it is a possibility, but it's not an easy possibility because the way the gene has been broken, essentially, it's not easy to resurrect that specific gene. And so it'd be it'd be easier if it was just, let's say, if it was there but turned off, it could be maybe easily turned back on again. But that's my dog shaking. Sorry if you can hear the jingling. But because there's actually been bits of the sequence totally destroyed, it wouldn't be easy for that to happen again. Obviously, that gene evolved once before, at least, so it could happen again. But it's probably more likely. It's a relatively unlikely scenario. So,
But
yeah.
we are still evolving.
Yes.
Yeah. Okay, that covers that then. Okay, thank you.
Tom, do you want to fire ahead?
Yeah, it's just a supplementary to that. You mentioned that the humans aren't unique amongst the upper apes and primates of having lost the ability to manufacture vitamin C in their bodies. I was wondered whether baboons and other species have a scurvy-like disease for want of vitamin C if they're deprived of it in the environment.
Yeah. I actually don't know that. I mean, certainly they would have the potential to have it. I mean, so I can't remember now which of the primates. I know it's definitely also gorillas and chimpanzees, but I can't remember where in the primate lineage off the top of my head that that gene was lost. So I can't remember if it's baboons, but let's say chimpanzees and gorillas, they would have the potential to have a scurvy equivalent. Whether that actually occurs naturally probably depends on what they're habitually eating. But having the absence, not being able to biosynthesize vitamin C, they would have the same problem that we would have in terms of if you don't have vitamin C, you have all kinds of deficiencies. I think the one that is the most obvious is the effect it has on collagen, which is why one of the effects they see is that the collagen totally breaks down, which is making up a lot of your skin integrity and these other various parts. But so it ought to happen, but I haven't read about it. It ought to be potentially possible, but I haven't read about it actually happening. That's
Thanks, Aoife.
Tom McLaughlin?
Yeah, Aoife, good evening, everyone. Just I asked a question there about CRISPR-Cas, the new gene editing techniques. And in 2018, the European Court of Justice basically said that gene editing techniques should be regulated like GMOs. And of course, as David McConnell said many, many years ago, the EU will bury our heads in the sand, perhaps regarding GM technology. And will we do the same again, like with CRISPR-Cas? How important is CRISPR-Cas in your research for medical uses? And I know it's important for plants, but this is something that can be really important from an Irish point of view and also from an EU point of view. Thank you, Aoife. A great lecture, by the way.
Thank you, Tom. So I'll say, so in my specific research in my lab, I'm not using it, but I can talk generally that I know that my colleagues are using CRISPR-Cas9 technology for almost anything. So not necessarily as medical therapy, not necessarily the case, but it could be that it's being used as a research tool. It's a really, really, really valuable research tool. It can also be eventually incorporated into medical therapies, but it is a very important tool in developing many fundamental understandings, because a classic way of understanding things in genetics was always to make a mutation in the lab in a gene, say in a fly or in a bacteria or something like that, or in cells that could be human cells growing in a plate. And with CRISPR-Cas9, you can do it in a deliberate, targeted way, and not trying to just get, so it's really, really powerful. And I know my colleagues are using it a lot. So this law that meant it was basically more difficult for them to do this research. I think it was a short-sighted ruling, if that helps. David can probably give a much more elaborated answer at some point whenever we open up the conversation.
Ian, do you want to fire ahead?
Okay, thanks. I had a little bit of a hiccup there. Aoife, I just wondered, about categories and about difference in kind, and just how do we distinguish species at a genetic level?
It's actually not that easy, and it is like a perennial problem. So you will find people arguing indefinitely on this, and maybe doing a whole PhD on like how do you actually define what a species is. So there are the easy ones, you know, which are easy to define. So you get past a certain amount of difference, and that's easy. So, and there are there's a concept which is called the biological species concept, which essentially just says that if these are capable of interbreeding successfully, then they are the same species. So successfully means that the offspring are also fertile. So a horse and a donkey can breed and they produce a mule or a hinny, depending on which one's mum and which one's dad. But those are sterile. So that so that is convincing evidence that those are different species. But that's easy. It's easier when they're so different. The challenge is when they're less different, how much difference is enough difference? And part of the problem there is that speciation is usually a gradual process rather than a single one-day event. So there are going to be different degrees. There's different stages along that process. So you might be witnessing what they might call incipient speciation, so something that's on its way to becoming a new species, but it could even still be reversible at this point. Like you can go, they could be getting different from each other. So two groups might be getting different from each other, and that if this continues, yes, they will be different species. But it could be still that they could get back together. Or you can go to past a certain point where it's no longer reversible. So it's a very, very difficult problem. And there isn't a single metric that tells you the answer, because you could get a genetic incompatibility between two groups based on a potentially small amount of difference, but it could be a large accumulation of lots of difference. And that can happen in different ways in different times and places. So there isn't one answer. There's not one rule.
Thanks. Khalid, do you want to go ahead?
Yeah, thank you very much, Aoife. It's a great pleasure, actually, a very nice lecture. I'm always concerned about this example of nature and nurture. And you have mentioned the example of the diving and the sickle cell anemia. And as acute medical physician myself, I deal with some practical uses of genetics in my daily life situation, and wonder about dating of a specific gene. For
dating,
example,
sorry, did you say dating?
Yes.
Yep.
Like dating as by age.
Mm-hmm.
So can I say this gene started to exist in the human species at this point of history, and it's been introduced because of such a kind of factor, whether it is nature or effects from other species or some weather changes? And can I also find out a mutation that happened at some point of history because of such intervention between the species, the human being, and nature? Thank you very much.
So kind of, yes, you can sometimes. I mean, so sometimes you can trace the origins of a particular mutation. You might be able to get a fairly accurate date on when it arose, but not always. And sometimes you can find a pretty plausible explanation for the circumstances that meant that new mutation then rose in frequency. So, but we can't, so it rising in frequency to become the dominant one, like so, or at least at a high frequency, like the example of the sickle cell anemia. We understand why that rose in frequency. We see the advantage it gives. We don't always have that information though. So so sometimes you can, sometimes you can't. And sometimes it takes a lot of work to do it. So one of the ways you might know how old a particular mutation is is by looking at how common that mutation is around the world. So if something is very geographically restricted, you could make a fairly justified assumption that it's relatively recent and it's just in, you know, maybe a big, like an extended family group. If it's very broad around the globe, it looks like an older mutation that has had time to move as those individuals moved and things like that. Though even saying that, there is a phenomenon where you can get essentially the same mutation happening independently in different parts of the world. And that's because some parts of the genome are a bit more prone to mutation. So what looks like the same event can happen in one individual and happen again in another individual, a bit like trisomy 21, you know, getting an extra copy of chromosome 21 happens independently each time. So you have different versions of that, as in smaller duplications, for example, can happen independently. So we don't always know why that a particular new mutation rises in frequency, and it doesn't always have a good reason. So one example, I think, that is evolutionarily interesting and medically relevant is the one that's called, you might have heard of it, Ccr5 Delta 32. So the cytochrome 5, is it cytochrome? No. Anyway, it's cytochrome receptor 5, and Delta 32 is a deletion of 32 letters from the middle of it. So this particular version of, so there's CCR5, the normal one, and there's the Delta 32 one. The Delta 32 one is associated with resistance to HIV infection. So that I
was very disappointed when I heard about it in college. When there is certain people, they can carry the HIV, but they cannot show any AIDS manifestation.
Well, basically the virus doesn't survive in their bodies because it can't. What happens is when HIV enters the T cells, it's binding onto this chemokine receptor 5. It binds there, and that's how it docks and enters the cell. So people who have this mutant version of that protein, HIV doesn't dock and bind, so it doesn't get into the cell. So it just dies out again quickly. But this was very interesting when this was discovered. It was discovered by accident by doctors in France, I believe, who noticed that certain individuals, despite living what you might describe as high-risk lifestyles, you know, sharing needles and things like that, were not getting infected, even though their friends were getting infected. And they looked for what was different in these people, and they found that they had this particular mutation. So this looks like, wow, is this some evolutionary adaptation to
a privilege?
HIV, except it's not, because you look at this and you see this particular mutation is much older than HIV. HIV is a relatively new virus that infects humans. It came from chimpanzees. We know roughly when HIV entered human populations, and we can see that this CCR5 Delta 32 is much older than that. So then people were going, well, is it something else? Was it another protection that this gene gave, this particular variant of the gene gave? And so one idea was, well, maybe it gave protection to plague or maybe it gave protection to smallpox. And all of these different ideas were explored, but none of them really looks convincing. So it looks like it's just a fluke. So even though this is a very medically relevant mutation, it's probably just one of those things, a bit like blood group variation. It's just one of those variants that exists, and it's there because of genetic drift. So anyway, so you have these stories like this.
It's very interesting, actually, and I'm personally interested in them, and looking forward to see which genome is more prone to decay by time and give out to some medical conditions which can be preventable. You know, so thank you very much for that. Very interesting.
Thank you.
Thanks, Khaled. I think just to put a limit on it, we don't want to keep Aoife taking a lot of your time this evening.
Well, I mean, I'll tell you when I'm ready, because maybe you're trying to politely tell me to go, but I mean, I've got nowhere else to be. It's locked down.
Like the next
At some point, yeah, maybe we'll say maybe like a few more minutes. I will go to.
We've got 3 more questions lined up. The next one is
Sure.
from Seema Sharma, and then we'll go with Ono Lynchig and then Ben Quinn, and we might then call it a day. Hopefully around, or who knows, but in the next.
Depends on how long I talk for each of my answers.
No, take your time. Seema, do you want to go ahead?
Yeah, hi. Thank you. It was great session, and I'm really happy I was been able to join from India actually, and it's here. Yeah, it's here 2:43. So it's quarter. It's going to be 3 p.m. here. Sorry, a.m. But today is Darwin's, day. So I just wanted to ask this particular question, which is there in my mind since ages, that why Homo sapiens, there is a single thing, Homo sapiens for male and female, why there is no categorization? Is there not sufficient genetic difference or whatever the criteria is? Because from my understanding, being a science person, I think that there is a huge, difference where women or female can get some genus and species, so I need your help to make myself understand.
Sorry, yeah. I think I need you to ask the question again, because I'm not 100% sure. So you're asking about categorization in humans?
Yeah, because Homo sapiens, Homo sapiens means, strong men, sorry, wise men.
Thinking,
wise men. So what about the women,
Okay.
And that is one thing. And the second thing is that the female, there is a difference, there is a genetic difference in male and female. So I think if you can, give Homo, genetic species name different to the different categories, and why male and female, they don't have, what is that? Why it is that?
I think I understand your question. So in a species, you would always give the males and the females the same name. So you wouldn't separate them out into different species, because they, they go together in that sense. When we talk about a species as something that's interbreeding, then they are all one species. So I suppose in that sense, the Homo, it's like saying man in the same way that Darwin said it in his book. It's just meaning human. But so yeah, you in any species, you group the males and females, which is not to say that there aren't differences. Of course, I mean, the most obvious genetic difference is the X chromosomes, 2 X chromosomes in the female and an X and a Y in the male. But that is a genetic variation the same way that any other genetic variation is. It's just one of the possible variations. It doesn't make them separate species.
Okay, but there is a bias in the name itself. So I guess there is some, it should have been something then, because I feel that, this thing need little more justification, because it's not very justified, which I don't want to get into that conversation, but this really sounds very, not very suitable, So, but thank you very much for your response. I was expecting this kind of response only, but I'm happy that, Iman Murphy took my question and I got my response.
And thank you for staying up so late to watch the
Thank you so much. Yeah, great. Thanks. I'm done. Thank you.
Thank you.
Owen, do you want to fire ahead?
Yes, can you hear me?
Yep.
Yeah, my question may be totally irrelevant. Thank you very much, by the way, for a very interesting talk. You spoke about the brains and similarities between the brains of all the species. Now, there is, I think, a difference in the level of consciousness between the species. They call it the hard problem. Do you have any opinion about the origin of the consciousness?
I think it's a hard problem. Yeah, no, I'm afraid it's, I think it's a fascinating question, but I don't think I have anything very clever to say about that, unfortunately. I think it is worth being aware, or just being conscious of the fact that we are a very egotistical species, so we know more about ourselves and what our brains are capable of and the way we can think. And, we possibly underestimate somewhat what the other animals are capable of. And I think, it's, Jane Goodall's work with chimpanzees, where she changed how people think about what chimpanzees are capable of, and people were very confident that they weren't capable of any kind of communication or tool building and things like that. But yes, I mean, clearly our brains are, we're capable of imaginative thought and all of these things that I don't know that other animals are capable of. But I find it an extraordinary problem. I find lots of things extraordinary problems in terms of, how can such a thing be encoded in the DNA, which it self-evidently is encoded in DNA, but I can't get my mind around that, how that is so. But yeah, sorry, I don't have anything better.
I think you're not the only person who can't get his or her mind around it.
Well, at least I'd take comfort in that.
Thank you very much.
Thanks.
Thanks for that, Owen. Ben.
Hi, Aoife. Thanks very much. That was really, really interesting talk. My question just related to a documentary I saw quite a number of years ago now. And I think they talked about two very isolated populations. I think one was in Iceland and the other in a valley in Switzerland, where they had suffered famine quite a number of generations ago, but they continued to remain pretty isolated in terms of breeding and that, and people, they didn't mix too much with outside populations. And according to this documentary, at least, they were finding that damage done to genes that they reckon the damage was caused by famine, that this persisted from one generation to the next. Now, I don't know whether that was just a theory being advanced and whether that theory has since been discounted or whether there's any truth in that. And I often wondered in terms of famine here in Ireland, I think is it in the west of Ireland that cystic fibrosis is a much higher occurrence, I think, than in Western Ireland than in many other parts of Europe even. I think it's cystic fibrosis, I might be wrong on that, but I just wondered, had you any comment on that at all?
So with regard to the famine thing, I'm not sure I heard the exact example you heard, but I did hear a similar story that was supposed to be in Dutch people who lived through World War II and experienced an extreme famine. And there was evidence that there were some genetic changes that apparently persisted for a couple of generations. But I haven't tracked that literature and paid attention to that literature very, very attentively. But what I remember was that the explanation was that if, for example, let's say I lived through World War II as a female living in World War II, and I experienced famine. As a female, my ovaries contain all the eggs that I will ever have from birth, right? I don't produce them later. So the ovaries were there and potentially are affected. And then any children I have were then potentially affected. And if they, so there was that potential effect like that, rather than it being a more durable effect over more generations than that. So that's the way I heard about it. But as I say, there could be more to it than that, because I haven't been tracking that literature very carefully. With regard to cystic fibrosis, yes, so we have a very high frequency. I think in Ireland, we have the highest frequency of carriers of mutation so in the gene, which then can, if somebody carries two of those, they will have the condition. And there's other conditions which are kind of peculiarly high in Ireland. Another one is hemochromatosis, which again is another genetic condition, which leads to abnormally high iron retention and which can accumulate and cause problems. And in both of those cases, we don't have a clear explanation for why it would be that those are high in Ireland. And it could be just one of those things. So with respect to Europe, Ireland is kind of at an extreme. And if you remember the idea I had of that serial founding effect, so you have a group and some of them move and some of them move. And so one possible explanation is just of the people who migrated to occupy this island, you know, if one of them carried a gene for this particular mutation that causes cystic fibrosis, and you know only 100 people moved and one of them carries it, then all of a sudden, you know, what might have been a 1 in a 10,000 frequency in the starting population is now a 1 in 1,000, or maybe it's a 1 in 100, depending on how many people move. So you get that kind of chance event, and that could be the same for hemochromatosis as well. There's another possibility that's been mentioned for cystic fibrosis, but I don't know if the evidence is very strong. But the idea is similar to the one to do with the malaria and the sickle cell, in that having one faulty copy might have an advantage under certain circumstances. And the potential circumstances were actually that it might give some resistance to TB. And being a traditionally farming culture, people have encountered TB through bovine TB. So that's an idea. It has merit as an idea, but I don't know if it's been really demonstrated. But that's one potential idea, is that the cystic fibrosis, the mutation that causes cystic fibrosis, when you have one of those and one ordinary copy, that you would have this advantage in terms of potentially resistance to TB. But like I say, I can't say for sure that that's true, but I think it's an interesting idea. So hope that helps.
If we have a people
Yes, thanks very much.
who are dancing their virtual arms by seeing if they can squeeze a question in there, would you mind taking 2 more from
Sure,
Robert?
yeah.
Sorry, may I be the one to ask a question? Thank you very much for the lecture, by the way. It was terrific.
Thanks.
I'm a geneticist, but I'm a scientist. But one question I would like to ask is, what is the main cause of mutations? And also, are viruses particularly susceptible to these because they're single chain, unlike DNA where the chains are locked together? And a little supplementary, I mean, what fraction of these mutations tends to improve the chances of survival?
Okay.
In other words, be useful to the viruses.
Yeah. So in terms of how mutations happen, in general, mutations happen when you copy the DNA, or if it's the RNA. So it's during the replication process. So for humans, the only time that matters is the replication that produces either the sperm or the egg cell, because so when your body grows, you can get mutations as well. And if I was to sequence the DNA from a skin cell on your left hand and on your right hand, I would probably find some differences. Very small amount, right? But I would find some, because every time a cell replicates, the DNA has to be replicated for that too. And every time you do that, there's just a chance that a mistake occurs. But in our cells, we have a lot of error checking mechanisms, DNA repair mechanisms. In fact, the Nobel Prize went for DNA repair, understanding DNA repair a few years ago, because it went to a few different people. And so even when errors occur, they can be reversed, and there's certain mechanisms to do that. So in a virus, even if it's some viruses only have a single strand, like you said, the RNA viruses, some of them are DNA, but they don't have the error checking. So they tend to, so the RNA ones evolve even faster, because the most basic error checking is at least that the 2 sides match each other and go together, because the 2 sides are complements. So DNA has that as its most basic kind of error check. But with RNA, which is only the single strand, there's not even anything to compare it against. So they change faster. In terms of how many of these are beneficial, so in the context of a human genome, yeah, I would say on average 0%, which is, so we got these 3.2 billion letters of DNA. And it's basically the idea is it's much easier to break something than make it better. If you throw a hammer at a car engine, you might improve it, you might, but more than likely you're going to break it. But with the viruses, they take advantage of the laws of large numbers. So viruses replicate they make so many copies of themselves. Even if the number of changes, the changes that might, well, so there's another thing as well, which is their genomes are much more compact. So our genome contains a lot of stuff that doesn't matter. So you could even cut it out entirely and you wouldn't notice. But the virus genome is the bare minimum. So almost all of it is doing something. So almost any change is going to have an effect, but it may be for better or for worse. So then they employ the laws of large numbers in that sense, because they make so many copies of themselves that they you can think of it like exploring all of the possible space, which is why when you have countries with huge numbers of viral infections, it's not a surprise that that's where we find these interesting variants of concern. But basically it's all down to relatively simple probability. But when you get lots and lots and lots of numbers of viruses, then those very improbable things happen. And even just to take the current case, one of the mutations that people are very interested in is this one that's been called E484K. So this is just a numbering system. So the number is the position in the protein. So it's in the spike protein. So 484 is the position in the protein, and an E has changed to a K. And those letters just symbolize the amino acids in the protein. So it's a glutamic acid became a lysine. This happened in South Africa, it happened in Brazil, and it happened in the UK now most recently. So the UK variant has now got a new mutation on top of it in some individuals, which is this new, so this mutation, the same thing happened independently in 3 different parts of the world, which is really improbable, but there's so many cases, so many viruses, and every time they replicate, they can mutate. So even though this apparently rare event happened in a very short period of time, independently in different parts of the world. So that's kind of what happens.
Thank you very much.
You're welcome. I can't hear you, Eamon. I can see your lips moving.
Sorry. Before we go to the last question of the evening from Peter Boylan, one of our members, Keane Harley, noticed that what appears to be a necklace representing healthy red blood cells.
Well, yes, okay, that's an accident, but yeah, okay, I'll pretend it was planned.
I had to ask. Peter, do you want to fire ahead with the last question? You need to unmute yourself.
Thanks, Aoife. That was a really fascinating lecture. And I just really have a comment that I think CRISPR is probably going to become an established practice in medical practice in hopefully the not too distant future. And it's kind of basic research like you guys are doing that is going to facilitate the development of that advance. And so thanks again. It's just a comment. It's late in the evening, so I don't want to delay anybody too much. Thank you.
Well, thanks very much. And yeah, so it's my wonderful colleagues who are doing that kind of work. And yeah, so there are some of my colleagues in genetics in Trinity that are actually developing genetic therapies, which are based on some CRISPR technology. Yeah, and it's extraordinary. It's such exciting stuff, and it's so precise. It's like because it is described as editing, and the metaphor is very apt. I think it is like you have a typo, and you can fix the typo. And it's such a precise technology that can really get to just fixing the problem and not doing anything else. So yeah, it is extraordinary. And I think it will be very important in medicine. I agree, the whole idea of personalized medicine. So I also admire that.
Good, thank you.
Thanks very much.
Thanks, Peter, and thanks, Aoife, as well. I think rather than me saying any more words about it, I think just reading some of the comments from the chat box kind of sum up the thoughts of the attendees, almost 90 of whom are still here 2 hours after we started.
I know.
Enthralling that was. Rosie McAdam said, really interesting lecture, could have listened for hours, fascinating talk. There's plenty of complimentary comments. I'm not going to do them justice. I suppose it just kind of remains for me to say thanks so much to you for giving up your time. We're really appreciative of the effort that goes into preparing something like that.
There's a lot of parties to be here.
I suppose it's that we're at that point in the pandemic, of course, but all the same, to put the effort into preparing and delivering this, we're really appreciative, and have somebody who's held in such high esteem within the scientific community, ready and willing to come and speak to us as well, is great as well. And thank you as well to David for giving the opening address. Always a pleasure to listen to you. And thanks everybody for coming and for participating and giving your questions as well. Otherwise, it's just myself, David, Aoife, and Alan Tuffrey in a virtual room talking about genetics. And it's not that any of us would couldn't think of worse ways of spending our time, but it's great to be engaged with so many Humanist Association members as well. I think just to conclude, if we could all give Aoife another huge round of applause, virtual one.
Thank you.
And
thanks.
Hopefully we'll see you again at some point, Aoife. Thanks.
Yeah. When we're back out in the world again.
Yeah, fingers crossed it won't be on Zoom the next time.
Yeah. Thanks very much, and thank you. It's a pleasure, and thanks for the invitation, and thanks for the amazing audience who stuck through that. I didn't realize I spoke for so long, so I can't claim it's uncharacteristic though.
You're very easy to listen to, don't worry.
Thanks very much.
Thank you.
Okay.
Night, everyone.
Nice. Thank you.
Bye.